Thank you very much for inviting me here to talk. I work in London. I'm training to be a gastroenterologist. My other interest is I work for a research registry of twins and multiplex families where one or both have inflammatory bowel disease. So we're both going to talk to you today about who gets IBD and why. I'm going to start off by talking about who, running through who is high risk for developing inflammatory bowel disease, and then go on to try to answer the perhaps more difficult question as to why. We'll discuss heritability, whether you can inherit inflammatory bowel disease or not, looking at the twin and family studies. Mars is then going to talk about the genetics of inflammatory bowel disease. I'll then talk a little bit more about the environmental factors that are known to be associated with IBD and the difficulties in interpreting these studies. And then we'll round up by discussing the interface between genes and the environment, such as the microbiota, which are the bacteria within our gut, and changes to our DNA that happen within our lifetime. So inflammatory bowel disease is an increasingly common disease. About 4 in 1,000 people within the UK are now affected. The IBD standards last reported that up to 620,000 people within the UK are now living with these diseases. So people of all age can suffer from inflammatory bowel disease. The most common age to be diagnosed is between 15 and 30 years of age, with a second smaller peak in IBD diagnosis between the ages of 50 and 80. Unfortunately, however, the incidence of new cases in children is rising. In Scotland in 2012, there was a large population-based study that showed the new cases have doubled over the last 20 to 30 years. Men and women are both affected by inflammatory bowel disease at about almost equal ratios. Traditionally, inflammatory bowel disease was thought to be a disease of the West. This map shows the countries affected by inflammatory bowel disease, colour-coded as to who's the highest, who have the highest numbers of IBD sufferers. And it can be seen that still inflammatory bowel disease is very high amongst Western Europe and in North America. However, all countries have people affected by IBD, and some areas, especially Asia, that were typically thought to be of low incidence, are now the diseases are rapidly on the rise. So does anyone know where this is? Anyone shout it out?
Iceland.
Not bad. Well, it's the Faroe Islands, where it is the highest chance that you'll develop IBD if you live there, with 80 per 100,000 people diagnosed with new cases per year. So IBD was also previously thought to be a disease of rich white people. It seems to be or highly represented in Jewish populations as well. However, some re-evaluation of the literature and some of the newer studies show that this isn't such a strong trend as previously thought. In particular, the North American studies, it was perhaps that poorer black communities were not being included in the data because they didn't have access to proper healthcare. One question that we're often asked is whether inflammatory bowel disease is heritable. Heritability is the extent to which a trait, in this case Crohn's disease or ulcerative colitis, is caused by our genes. This isn't that straightforward to answer. There are some ways in which heritability can be calculated. One is by looking at family studies, another is looking at twin studies, and a third way is by looking at the genes that we've so far identified that are linked to inflammatory bowel disease and estimating what proportion of the IBD cases diagnosed these can account for. So looking at the family studies, the greatest risk factor that anyone can have for developing inflammatory bowel disease is having one or more close family member diagnosed. A sibling of someone with Crohn's disease has up to 40 times more chance of developing Crohn's disease in their life than a member of the population with no relatives diagnosed with inflammatory bowel disease. In ulcerative colitis, this is up to 15 times. However, there are many people that develop inflammatory bowel disease that have no family members affected at all. In fact, the majority of cases, about 85%, have no known family history of the disease. Inflammatory bowel disease in families tends to also be similar in behaviour, location, and the age of onset. It also, unfortunately, tends to present earlier and follow a more severe disease course. However, when interpreting the family literature or the family studies, it's important to remember that families don't only share the similar genes, they also share a similar environment. Twin studies allow us to control a little bit for shared environmental factors. They start with the assumption that identical and non-identical twins both share a roughly equally similar environment, especially in early life. We know that identical twins have identical genetic code, and non-identical twins share the same genetic code as other siblings, which is half as similar as that as identical twins. Twin studies identify twin pairs where one or both are affected by inflammatory bowel disease. They then calculate the concordance of the twin pairs, and that means whether amongst a twin pair both twins go on to develop the disease. It is the difference between the concordance of the identical and the non-identical groups that is key. And if there's a big difference between them, this suggests that the genetic similarity of twins, of the identical twins, is in part contributing to the disease. As an aside, identical twins will share 100% concordance for chickenpox, but so will non-identical twins. So just the fact that both of them get it itself is not enough to show that the disease is heritable. This slide shows some data from the UK Inflammatory Bowel Disease Twin and Multiplex Registry, which I've helped set up, and some other twin studies across European populations. The first slide shows our concordance rates for the first 100 twin pairs that registered with our project. Looking at the identical twins with Crohn's disease, about half of them both go on to develop disease after one twin has been diagnosed, compared with just under 10% of the non-identical twins. And this suggests that Crohn's disease has a strong genetic component. Looking at the groups with ulcerative colitis, the identical twins that we've got within our cohort, 25% of them both have disease, compared with just under 20% of the non-identical twins, suggesting that perhaps ulcerative colitis is less heritable than Crohn's. Now, Miles is going to come up and tell you a bit more about the genetics of inflammatory bowel disease.
Thanks, Hannah, for that great introduction to this topic. So 20 years ago, as a knock-kneed registrar, I was presenting in this same room. It was just occurring to me just now. I have a feeling that the stuff I was presenting was largely garbage, mainly because we didn't really know how to do genetic studies in those days. We didn't have the technology, and we hadn't worked out that you need really big panels of cases, i.e. patients, to really make these studies work. Now, many of you may have contributed to some of the studies that we're now going to talk about. What do we do with our genetic studies now? We do what's called an association study. And in an association study, we essentially compare the genomes of people with IBD to people who don't have IBD. And what we're trying to do with these studies is to identify gene variants which are associated, so-called, with inflammatory bowel disease. Why would you want to try and do that would be one question. And the answer to that really is, if we can work out what these gene variants are doing, we can understand more about the pathways, the molecular pathways and the biological pathways that cause Crohn's disease and ulcerative colitis in the first place. And what we're hoping that might lead to is to a better understanding of what the environmental triggers are, and we'll be talking more about those in a minute, but also to directly identify some druggable targets. The key stage, we've done very well with regards to this identifying gene variants. I'll mention some of those things in a minute. The next step is critical, working out the functional impact of these gene variants and what they're actually doing to cause IBD. And this is where we're going to have a massive role for what we call the IBD bioresource, something which Crohn's and Colitis UK and James Pryor, who's on the stage here, have been absolutely instrumental in getting going and have allowed us to attract more than £1 million worth of funding from the MRC and the Wellcome Trust to establish this bioresource. I can talk more about that later on if you'd like me to. Genome-wide association scanning then allows us to do an unbiased survey of the entire genome. We do what's called genotyping on hundreds of thousands of markers in thousands of cases and controls. We get our clever statisticians to analyze all the data that we generate and locate these so-called SNP markers at which we see regions of association. And this is just a graph of the whole genome here, and where you see these sort of tower blocks of SNPs, these are areas which contain disease susceptibility genes for Crohn's disease or ulcerative colitis. We then see which genes map to those parts of the genome, and that then is the first stage of beginning to understand exactly how these things are causing Crohn's disease or ulcerative colitis. Now, as I said, 20 years ago we were no good at this, but in the middle of the last decade we got really pretty good at doing it, and since then have published a whole constellation of papers, and these are just a sample of those studies coming from mainly our UK IBD Genetics Consortium, which I've chaired now for 10 years or so, but also from the International IBD Genetics Consortium, which I helped co-found some years ago, and which has, since I've given up, has gone from strength to strength, I'm delighted to say, in a wholly good way. This is the latest output from that international consortium. You probably can't see the figures down here, but about 20,000 or so patients with Crohn's disease from around the world, not dissimilar number of patients with ulcerative colitis included in this study, and 30,000 or so controls. You can see the UK punching disproportionately above its weight here compared to the rest of the world, and that's again largely credit to you and the support that Crohn's and Colitis UK have provided. Now, in total, about 160 loci, which are showing discrete association with susceptibility to inflammatory bowel disease. This knocks spots off any other common disease, actually, in part reflecting the peculiarly heritable nature of Crohn's disease, but also the fact that through your efforts and everybody's efforts, we've been able to assemble much bigger cohorts of patients to help us to do these studies. So what have we learned? Well, firstly, that there's a huge overlap between genetic susceptibility to Crohn's and ulcerative colitis. Perhaps not a surprise, but interesting to see. And we can now, in rather granular form, understand the details of which of these loci are predominantly associated with Crohn's, which with UC, and which ones are shared, and what their relative proportion to that genetic susceptibility is. In some pathways that we've identified, you see a whole number of molecules in the same molecular pathway which are showing susceptibility or variants within which are showing susceptibility to IBD. And of course, these are major clues and are already being taken into through drug discovery programs and into clinical trials indeed. And in fact, some of the most exciting drugs that are coming through, new drugs that are coming through for IBD, are directly targeting this pathway, first highlighted by genetic studies. One of the things that's interesting is that the genes that we discovered early on have a bigger effect than the ones which are coming out later. This is so-called NOD2, which you may have heard about. But if you get 2 DUF copies of NOD2, then your risk of getting Crohn's disease is about 40 times people who don't have any copies of NOD2. Interestingly, the mutations in NOD2 cluster in this part of the gene product, which is to do with sensing bacteria. And that's going to be a theme that we're going to come back to. Another interesting story is the so-called autophagy story. This gene showed association, in fact, in 3 independent panels, more or less simultaneously. And in fact, in our study run from the UK, which was a rather bigger study than everybody else's, we also identified association at this second autophagy gene. And this nicely illustrates, I think, the power of this technique of genome-wide association scanning. Nobody had thought at all about this pathway as being remotely involved in Crohn's disease susceptibility before. But with the application of this new technology, we are able to identify these and, in fact, subsequently other genes in this pathway. And what autophagy does, it describes that within cells, it's the formation of these membranes that encapsulate, in fact, what in IBD terms are likely to be bacteria within the cells and target those for destruction by lysosome. And it looks to be there's some defect in this process which predisposes to Crohn's disease. In ulcerative colitis, a significantly overlapping set of genes, but interestingly, some which don't overlap. And these are largely to do with permeability, the leakiness of the gut, if you like. What we don't yet understand is why this should be ulcerative colitis specific and really what it is that leaks through. It's also been interesting to see the significant overlap between IBD genes and genes that confer susceptibility to a range of other inflammatory and autoimmune types of conditions. And also of particular interest, actually, from the scientific point of view, big overlap with so-called mycobacterial susceptibility loci for leprosy and this other mycobacterial disease down here. And as indicated at the bottom here, it rather implicates mycobacterial disease as, well, maybe not playing a causal role in IBD, but there's maybe some sort of evolutionary imprint of how our bodies have adjusted to mycobacteria or how they fight off mycobacteria that maybe puts us at increased risk of developing IBD in today's clean world. I'm going to hand back to Hannah now.
Hello again. Well, we've heard from Miles how important the genes are, but looking back to our twins, even with Crohn's disease in the highest concordant rate population, 40% of people with identical genes will not go on to develop inflammatory bowel disease, even though their twin has it. So clearly, something other than genetics is also vital. The rapid rise in inflammatory bowel disease, especially with what's going on in Asia at the moment, is also far beyond that can be explained just by the genetics. In Eastern Asian countries, there's been a 2 to 3-fold increase in inflammatory bowel disease over the past 10 to 20 years. The patterns that they see over there actually mirror what was seen in Western countries about 50 years ago. Right now, there are more cases of ulcerative colitis being diagnosed, although Crohn's disease is now increasing so rapidly that this trend is likely to level off pretty soon. There are less cases of family history reported amongst Asian populations. And as I was saying before, this rapid increase far outweighs that could be explained by new genetic mutations. Migration studies look at what happens when people from low areas of inflammatory bowel disease move to higher areas such as Western Europe, Australia and Northern America. Essentially what happens is as time goes on, their risk of inflammatory bowel disease increases. In fact, in second-generation Asian migrants that have moved and built families and lives in Leicestershire, the ulcerative colitis rates are now comparable to that of the British Caucasian population. When people from Asian populations get inflammatory bowel disease, it's generally as severe, if not more so. It's still more likely to affect men, and it's less likely to affect anywhere that isn't in the gut. So why has this increase happened if it's not due to our genes? One explanation is the westernization of our lifestyle. In particular, diet has been implicated, which I'll talk about in a little while. It's thought that perhaps better sanitation and hygiene, especially during our formative years when our immune systems are developing and when the bacteria within our gut are developing, may have a critical role. It's possible also that infection is implicated. This slide here shows all the environmental factors that have been linked with inflammatory bowel disease. Looking at the medication, some studies have shown associations with aspirin, anti-inflammatories, the oral contraceptive pill, and more recently in the Daily Mail, antibiotics. Lifestyle factors associated include smoking, which is predictive of Crohn's disease but protective of ulcerative colitis, diet, low exercise, and stress. Health situations involved that may be precursors to inflammatory bowel disease include high reports of childhood illness, gastrointestinal infection, possibly atypical mycobacterium, and previous history of appendectomy. Our early environment is likely to be important. There's an association with emergency caesarean delivery and subsequent development of inflammatory bowel disease. The same has not been replicated with elective caesarean deliveries. And then some studies show an association with breastfeeding being protective, although other studies show infant feeding makes no difference whatsoever. As for specific dietary factors that have been implicated, the studies show slight variations, but the main themes are as follows. Prepared ready-made meals are thought to be bad, as are polyunsaturated fats, high sugar, low vegetable, low fruit, and low fibre diets. Interpreting these environmental factors research is tricky. Firstly, the papers look at correlation, and for the vast majority of environmental factors, we don't have clear mechanisms between cause and effect. Secondly, as I've said, the results vary considerably between studies. For example, looking at the antibiotics, antibiotic use has been associated with paediatric cases of inflammatory bowel disease in the UK, whereas Asian studies have shown the opposite. Each individual risk factor on its own may, across the whole population, confer a risk for inflammatory bowel disease, but to each individual the effect is relatively modest. These risk factors are often not modifiable. Going back to the antibiotics, people often usually prescribed antibiotics because they need them. And finally, I think it's very important to avoid attributing guilt or blame to why people get these diseases. The last thing we want is anyone thinking something they may have done in their pregnancy, in their early life, or their 2 cigarettes that they smoked or 200 cigarettes that they smoked have caused their disease, because it's not useful or helpful to think in that way. Miles is going to talk a bit more about the microbiome.
Microbiome in 2 minutes.
Yeah, sorry.
In fact. No, that's good. So now that Hannah's woken you up from my little genetic sojourn, I'm going to explain to you a little bit about the microbiome, which is very important. As you're all aware, we're all full of bugs, 10 times more than there are cells in our own bodies, and they're part of us. They're very much part of us. And you can think of IBD as an autoimmune condition where the target, rather than being the thyroid or your pancreas for diabetes, is the microbiota. And it's important in a whole constellation of different disorders relating to medicine. We all vary a lot in terms of the bugs that we have within us, and this is a sort of list of the species or taxa that there are. But interestingly, the metabolic pathways of these bugs are actually quite stable between individuals. They're influenced by a whole constellation of different things here, which I'm not going to go through in detail. What we do know is that the bugs within us profoundly affect the immune system in our guts, both in terms of how it behaves now, in adulthood, but actually in particular, how it develops through infancy. In IBD, in Crohn's disease, where there's more data than ulcerative colitis, there's a so-called dysbiosis. The numbers of bacteria or the richness of the bacterial populations are quite dramatically reduced, and there's reduced diversity, so-called. There's particular decrease in these things in green at the bottom here, and some species, such as the E. coli, are actually somewhat increased. And you can chase this through, and there's particular interest in F. prau, which seems to have some sort of protective role in Crohn's disease. There's a whole and burgeoning literature on the bacterial contribution to human health. More recently, we've actually looked at viruses as well. This is a collaborative study I've done with a big North American group, and shown that in contrast to the loss of bacterial diversity associated with IBD, you get a bloom of viruses that affect these bacteria. And we're just drilling into exactly what's going on there as to see whether those two things are correlated directly. The microbiota and IBD, it's unclear at this stage whether the changes that we can observe happen before the IBD starts and are somehow triggering that off, or are a secondary response. And it's very difficult to tease out this causal correlation. What we do know is that we can change the microbiota. Diet profoundly affects it, and some of the changes which you can see in healthy people as they change from meat to a vegan diet are mimicked by IBD. Smoking affects it. But we do know, at least in the preliminary studies that have been done so far, prebiotics and probiotics and dietary modification don't obviously play a clear-cut role, at least, in settling down IBD. Just one slide on epigenetics, and then the summary slide. Epigenetics sort of describes the programmed regulation of gene expression. It's a way the environment can sort of get its own back on all those geneticists. And you end up with a situation whereby the environment can influence how genes become transcribed in quite a stable way that's then transmitted as cells divide. There's an increasing literature on this relating to IBD. I'm not going to go into any details. So in conclusion, as Hannah has said, anyone can get IBD. There are a number of risk factors that have been identified, and all of these are being investigated at the moment. What we're hoping is that by better understanding why people get IBD, of course, we're going to be able to do something about it. And just a conclusion slide, just to list some of our funders, and in particular, obviously, Crohn's and Colitis UK. Thank you very much indeed.